My pleasure to introduce now Omer Yilmaz, who's an assistant professor in biology here at MIT and in the Koch Institute. He's also a gastrointestinal pathologist at MGH and HMS. And perhaps excitingly was just named as a Pew Stewart Scholar for Cancer Research just yesterday. This is an award given for young investigators that are at the beginning of their careers that are doing very exciting work in the space of cancer research. And so he's going to tell us today a little bit about his work on dietary control of stem cells in obesity and cancer. Omer?
Thank you, Chris, for the introduction. And it's a pleasure for me to present some of the work that we've done in the lab trying to understand how obesity contributes to the early steps of intestinal tumor formation. And that's a really important question because if we can identify changes that occur in normal tissues before they acquire mutations that give rise to tumors, one might be able to identify biomarkers that can identify populations of patients that are at more risk of developing cancers. So in my lab, we're really interested in understanding how diverse diets impact adult stem cells in the intestine and how these changes in adult stem cells contribute to tumor formation. So in the lab, we look at low-calorie diets such as caloric restriction and fasting. And the reason we look at these low-calorie diets is because caloric restriction, for example, is well known to have anti-tumorigenic effects. It reduces tumor initiation, it reduces tumor progression. On the other side of the spectrum, my lab is really interested in understanding how pro-obesity diets, such as a high-fat diet, Western diet, high-glycemic diets, how these pro-obesity diets alter adult stem cells and increase their propensity to give rise to tumors. And ultimately, we're really interested in understanding how diet has a strong effect on aging and diseases of aging, such as cancer. So as has been mentioned in many of the sessions today, obesity is a modifiable risk factor for cancer incidence. So we've known for the past decades that indeed obesity or increased body adipose correlates with with cancer incidence in multiple tissues. So for example, in a tissue that I'm going to talk about today in colorectal cancer, patients that have a BMI of greater than 30 have a modest but significant increase in the incidence of intestinal cancers. Not only does this correlation hold in the intestine, but it's also true for many of the organs in the gastrointestinal tract, such as the liver and the esophagus. Not only are these changes restricted to the gastrointestinal system, but obesity correlates with increased cancer incidence in other tissues, such as breast and endometrial carcinoma. So the question that we're really interested in is, what is the role of diet and obesity in the early steps of intestinal tumorigenesis? So we're really interested in understanding how does normal mucosa in the intestine, what are the changes that occur in normal intestine in obesity that eventually acquires mutations and gives rise to invasive cancer? And we think the intestine is a great system to study thanks to pioneering work by Bert Vogelstein and many others, because we now understand many of the early drivers and late drivers of intestinal tumor formation but we know very little about the changes that occur in the normal mucosa in response to obesity. So we're really interested in understanding how, for example, a pro-obesity diet, how does a pro-obesity diet influence tumor formation at these various time points in evolution? And today I'm going to really focus on the changes that occur in obesity in the intestinal epithelium. So I'm going to really focus on adult stem cells because adult stem cells are often the initial cells that acquire mutations that give rise to frank cancers. And work by Vogelstein and others have shown, for example, that there's a very strong relationship between total adult stem cell divisions and cancer risk. And our model system for studying this, as I've mentioned to you, is the mammalian intestine, where in the intestine, one has these intestinal crypts, which are the workhorse of the epithelium. These intestinal crypts house all of the adult stem cell populations, and work from the Cleaver's lab has identified a marker, LGR5, that exquisitely identifies these adult stem cells in the intestine. called LGR5. And using a genetic approach or using in situ hybridization, one is able to identify these LGR5 positive cells. So using a genetic approach, when one knocks in a green fluorescent protein into the promoter of LGR5, the LGR5 positive stem cells are green, and these green LGR5 positive stem cells reside at the bottom of these intestinal crypts. These LGR5 GFP high stem cells then give rise to more differentiated non-stem cell progenitor cells or transit amplifying cells, which are GFP low. And these GFP low progenitor cells give rise to the many different cell types of the intestinal epithelium. And it's important to note that in the intestine, these LGR5 positive stem cells are adjacent to Panna cells in the small intestine and in parts of the colon. And in the remainder of the colon, they're next to these deep secretory cells. And these niche cells, these Panna cells and deep secretory cells, are really important for regulating the biology of these LGR5 positive stem cells. So in the intestine, like many adult tissues, there's a hierarchy where you have these rare adult stem cells which reside at the apex of the hierarchy, which give rise to more differentiated and more numerous cell types. Another reason why we're really interested in focusing on the intestine is because one has many robust assays to really dissect out how a given intervention regulates stem cell function. So one, for example, using flow cytometry and these LGR5 GFP reporter mice, one is able to prospectively isolate out these stem cells and these niche cells and co-cultures them together in 3D matrix and assay the ability of how a given intervention regulates the biology or the ability of these stem cells to give rise to these mini intestines or mini organoids in culture, shown here. So here's a schematic of what these mini organoids look like. They have these cryptomains and these pseudovillus domains, and this morphology is very reminiscent of the morphology that one observes in the mammalian intestine. And here's an example of an organoid derived from a single LGR5 GFP positive mouse, and one can appreciate that these GFP positive stem cells, just as they do in the in vivo setting, reside at the base of the intestinal crypt. And this is a really powerful platform because my lab is really interested in understanding how different dietary constituents, for example, of the high-fat diet or in other dietary types, alter stem cell function in a controlled ex vivo setting. So now I'm going to talk about some recent work that we've done in the lab, trying to understand how one model of obesity alters stem and progenitor cell biology and how these changes augment cancer incidence. So our model for inducing obesity is a high-fat diet. These mice acquire 60% of their energy from fat or lard. And we age these mice for at least 1 year on this high-fat diet. And we aged these mice for at least a year. So here's the high-fat diet and its control. It's quite obvious that these mice are obese. And interestingly, if one assesses cancer incidence in these high-fat diet mice, one finds that these high-fat diet mice develop more spontaneous intestinal tumors, shown here grossly and microscopically, than their control-fed counterparts. So what is the mechanism? How does this pro-obesity high-fat diet impact adult stem and progenitor cell function? So we wanted to understand what are the quantitative changes that occur in adult stem cells and what are the qualitative changes that occur in response to obesity. So we use many approaches to quantitate adult stem cell numbers. So one example here is by using flow cytometry, where one enumerates the numbers of these LGR5 GFP positive cells in high-fat diet mice and in control mice, and one can appreciate that there's about a twofold increase in the pool of LGR5 positive cells in obesity, both in the small intestine and in the colon. So what about qualitative changes? In addition to having more stem cells, is the function of these stem cells different in a high-fat diet? And here we utilized the organoid assay, and we isolated stem cells and their niche cells from control and high-fat diet mice. So here we're looking at the ability of these stem cells to give rise to these mini intestines in culture, and here we're looking at, on the horizontal axis, we're looking at stem and Panna cells, or these niche cells, from the different dietary conditions. So control stem cells by themselves are ineffective at giving rise to these mini intestines in culture. However, when one co-cultures these stem cells with their niche cells, the presence of a niche cell dramatically enhances the potential of these stem cells to give rise to these mini intestines in culture. Importantly, what happens in a high-fat diet? In a high-fat diet, we make a very surprising finding, which is in a high-fat diet, these stem cells now acquire niche independence. So these high-fat diet stem cells are now as functional as these control stem cells that have been co-cultured with their niche, indicating that there's changes that are occurring in the high-fat diet intestine that's now uncoupling the function of these intestinal stem cells from their neighboring support cells. Moreover, if one co-cultures these high-fat diet stem cells with their neighboring niche cells, they can still respond to cues that are elaborated by the niche. And this is really exciting because it raises the possibility that one of the earliest changes that occurs in the intestine in response to a high-fat diet is this uncoupling of stem and niche cell. And not only do we see this phenomenon in the organoid assay, but actually when we look at sections of the intestine, we can appreciate that there's a clear increase in the numbers of intestinal stem cells and a reduction in their niche numbers, indicating that both in vivo and in vitro, there's uncoupling of stem cells from their neighboring support cells. So we then wanted to understand, we then asked, is it really constituents of the diet that are driving this phenotype, or is it sequelae of obesity that are driving this phenotype? And again, leveraging use of this organoid assay, one is able to address this question. And the question is, I've shown you that a high-fat diet induces stem cell self-renewal. Remember, in a high-fat diet, we have more stem cells, so they've undergone more self-renewal divisions. And the question is, can we mimic these effects of the high-fat diet in our controlled ex vivo assay by exposing naive organoids to palmitic acid? And why did we choose palmitic acid? We chose palmitic acid because palmitic acid is one of the dominant fatty acids found in the high-fat diet that these mice receive. And here's the data from that experiment where we took naive organoids and exposed them for 3 to 4 weeks with either vehicle or palmitic acid. One can appreciate that exposure to palmitic acid leads to more green, and remember, green is a proxy for LGR5 numbers. One can quantitate this high throughput using flow cytometry, and when one does that, one finds that there's a twofold increase in the number of green cells in the palmitic acid treated organoids compared to their vehicle counterparts. And importantly, when we look at the quality of these stem cells that have been exposed to long-term palmitic acid in the organoid assay, one finds that palmitic acid-derived stem cells are 3 to 4-fold more likely to give rise to these mini intestines in culture. So they've now co-opted the ability to more efficiently give rise to these mini intestines in culture, akin to what we observed in a high-fat diet, where in the high-fat diet, remember, stem cells acquire niche-independent growth. And we think that these effects need not be specific to palmitic acid because we're able to recapitulate a lot of these effects using other fatty acids that are commonly found in the high-fat diet, such as oleic acid, and we've used a commercial mixture that mimics the high-fat diet. Moreover, we don't think that this is something specific to mouse tissue because we're able to recapitulate these effects using patient-derived or human mini intestines. So what is the mechanism? So to address the mechanism, we performed RNA sequencing, and we identified one pathway that was dramatically induced in the high-fat diet compared to their control stem and progenitor cell counterparts, which is the PPAR-delta pathway. And these PPARs are nuclear receptors. They reside in the nucleus, and upon engagement of their ligand, which isn't well understood, but it's believed to be fatty acids or derivatives of fatty acids, upon engagement of their ligand, they drive a very robust metabolic program, which in this case for PPAR-delta is fatty acid oxidation, thus enabling these stem and progenitor cells in a high-fat diet, we think, to utilize fat as a mechanism for generating ATP. And we validated that indeed the PPAR-delta pathway is induced in the high-fat diet stem and progenitor cells. Here we're looking at protein levels in sorted stem and progenitor cells of CPT1A, which is the rate-limiting step of fatty acid oxidation, HMGCS2, which is the rate-limiting step of ketogenesis, and fatty acid binding protein 1. And one can appreciate that in a high-fat diet, both stem and progenitor cells induce these targets of PPAR-delta. Interestingly, PPAR-delta levels themselves don't change. And we've also verified this at the mRNA level. We've done in situ hybridization and IHC. So up until now, I've shown you that our working model is that we think that perhaps a high-fat diet and fatty acid constituents or components of the high-fat diet are activating a PPAR-delta program. And an important question is this PPAR-delta program sufficient, necessary, or both for mediating these effects of a high-fat diet? And the PPARs are great to study because one has a lot of pharmacologic and genetic tools that enable us to dissect out its role in this phenotype that we observe in a high-fat diet. So there's one agonist that one can use called GW501516. From here on out, I'm just going to call this GW. And this GW compound is a very potent and specific activator of PPAR-delta. So to address the question of is PPAR-delta sufficient for mimicking the effects of a high-fat diet, we administered this agonist, this PPAR-delta agonist for 3 to 4 weeks to mice in vivo, and then tried to understand how close does treatment with this PPAR-delta agonist mimic the high-fat diet. So interestingly, when one looks at numbers of stem cells in mice that are treated with GW here shown in black, just as we observed in a high-fat diet, with GW treatment, we observe almost a 2-fold increase in the total numbers of these LGR5 positive stem cells. In data that I'm not going to show you, we find that both a high-fat diet and an agonist activation of PPAR-delta boost stem and progenitor cell proliferation. And importantly, what about function? Does synthetic activation of PPAR-delta enable these cells to give rise to mini-intestines in the organoid assay independent of their niche cells? And indeed, that's the case. So here we're looking at, there's a several-fold increase in the ability of these GW-derived stem cells to give rise to these mini-intestines in culture shown here compared to their control counterparts. And lastly, we wanted to address the question of necessity. Is PPAR-delta itself necessary for driving these effects? So we've generated now PPAR-delta knockout mice. So when one takes PPAR-delta deficient organoids and exposes them to fatty acids in culture or to our PPAR-delta or to our PPAR-delta agonist in vitro, one is able to block the organoid enhancing effects of these fatty acids and our GW compound, indicating that PPAR-delta mediates fatty acid-driven organoid self-renewal. So now I want to circle back to why my lab, why we were interested in understanding. I want to come back to the beginning where we asked, why does obesity lead to an increase in intestinal tumor formation? And as I've mentioned to you, normal tissues are often maintained by adult stem cells, and there's a lot of data over the past decade implicating these adult stem cells as a cell of origin for many tumors in diverse tissues. So these adult stem cells acquire mutations. They give rise to either other stem cells or daughter cells that go on to give rise to frank cancers. But what could be happening in obesity? So in obesity, we think that what's happening in the intestine, at least, is that in a pro-obesity high-fat diet, one has more stem cells, shown here in red. And by virtue of having more stem cells, one has more substrate that are available for undergoing oncogenic transformation. We also hypothesize that perhaps non-stem cells in the intestine can undergo oncogenic transformation and contribute to tumor formation. So why do we hypothesize this? So up until now, I've really focused on the effects of a high-fat diet on these primitive LGR5 positive stem cells in the intestine. But what about other more differentiated progenitor cells in the intestine? So here we're assaying the ability of these non-stem cell progenitors to give rise to mini intestines in culture. As I've shown you earlier, control and vehicle treated progenitor cells are unable to give rise to these mini intestines in culture. However, those from high-fat diet or from GW treatment are now able to give rise to these mini intestines in culture, indicating that high-fat diet now confers organoid initiating capacity to these non-stem cell populations in the intestine. And this is really important because organoid initiation is a property that we ascribe to these LGR5 positive stem cells in the intestine. So we wanted to ask, can these non-stem cell populations in the intestine give rise to tumors in mice? So we developed a genetic model where we can delete a tumor suppressor gene APC, which is often mutated early on in intestinal tumor formation, and we delete it in stem cells and non-stem progenitor cells, and then we isolate these cells shortly after deletion of APC and transplant them using colonoscopy into the colons of recipient mice. And has been shown previously by Clears and colleagues, under normal physiology, only stem cells are able to transfer these adenomas to recipient mice. However, when we delete APC together with activating PPAR-delta using our GW compound, we find that both stem cells and non-stem cell progenitor cells can transfer tumors to recipient mice, indicating that PPAR-delta permits these APC null intestinal progenitor cells to form in vivo tumors. And I just want to summarize by saying that, we think that a high-fat diet PPAR-delta program enhances stemness and tumorigenicity of intestinal progenitor cells. Under normal dietary conditions, one finds that stem cells serve as a cell of origin for tumor formation, but we think an early change that occurs in high-fat diet is that not only can stem cells gain niche independence, these non-stem cells acquire stemness features, such as the ability for giving rise to organoids in our in vitro assay, and importantly, both stem cells and these more differentiated progenitor cells can contribute to the pool of cells that can undergo oncogenic transformation and can serve as cells of origin for tumor formation. And we think these data are really exciting because, like every exciting finding, they raise very exciting questions, such as reversibility. How stable is this mechanism when one comes off of the high-fat diet or when one becomes post-obese? Aging. We're now looking at alternate models of obesity, and we think, for example, mice that are on a more carbohydrate-rich diet actually have a different phenotype than this pro-obesity high-fat diet. And interestingly, I just have 2 or 3 slides left. We think that these findings have very important implications for the treatment of cancer. And we think that a therapeutic implication of these findings is that targeting PPAR-delta or its downstream program in the appropriate dietary metabolic context might be a way for treating cancers that arise in obesity. So I've shown you that a fat-rich diet or a mimetic of a fat-rich diet such as GW activates PPAR-delta and drives stem and progenitor cell activity, leading to tumor formation, raising the question of whether inhibition of PPAR-delta either early on or later on can attenuate or dampen cancer incidence and progression in tumors that arise in obesity. So what we're really interested in asking is, do tumors that arise in a high-fat diet become dependent on PPAR-delta for their maintenance? And indeed, it turns out that tumors that arise in a high-fat diet, just like their normal counterparts, like the normal tissue, these tumors that arise in a high-fat diet strongly activate a PPAR-delta program. So when we look at the 2 rate-limiting steps of fatty acid oxidation, which are well-known PPAR-delta targets, CPT1A and HMGCS2, we see very low expression by in situ hybridization in these tumors. However, in high-fat diet, we now see strong induction of of these metabolic downstream targets of PPAR-delta. So we wanted to test this functionally. If we delete PPAR-delta together with our tumor suppressor gene APC in normal diet and in high-fat diet, will we have a selective effect on those tumors that arise in obesity? So when one deletes PPAR-delta together with APC in normal diet, unfortunately, we don't see any extension. So PPAR-delta, these data indicate that targeting PPAR-delta in normal physiology might not be a very valuable approach. But what happens when one targets PPAR-delta in tumors that arise in a high-fat diet? The findings are really dramatic. So here one finds that a high-fat diet significantly accelerates tumor formation. So the mean survival decreases from around 80 days down to 30 days. And interestingly, when one now targets PPAR-delta in these tumors, one almost doubles the mean survival of these mice. So we're really excited by these findings because it suggests a proof of concept that it might be important to target the metabolic state or the physiologic state of the organism in which the tumor arises. So with that, I'd just like to summarize. We're really excited because our future direction is to understand how diverse diets impact intestinal tumorigenesis, particularly those early steps that occur in the epithelium before the acquisition of mutations happen. We want to define and understand different models of obesity and how these alter intestinal stem cells and cancer. We want to identify and exploit diet-induced metabolic liabilities. We're looking at the high-fat diet, ketogenic diet, Western diet. We're really interested in understanding how these targets of PPAR-delta might be exploitable in both mouse and human tumors. And we're now testing a lot of these ideas in patient-derived organoid assays. So with that, I'd just like to acknowledge our many collaborators in the Boston area, including members of my own laboratory, and thank you.